Hello, we're going to, or should I say, I'm going to talk about section 7.1 and 7.2. I know we talked a little bit about this in class. We're going to start with the phospholipid bilayer, and that would be two layers of, we have our phospholipid here. Ooh, that was awful. There's one layer, there's the other layer. It is going to act as a partition so that things can't easily go in and out. We don't want the cell to leak materials that it needs out. We also don't want things to come in that does not belong. So we have what's called the fluid mosaic model. I'm going to see if this will play because it's really short, but it is kind of cool. Let's see if I can get this to work. No, and I don't think I can. Oh, let's try this one. This might be it. Yeah, I was afraid it wouldn't play while I was trying to do a presentation. But if you have time, do watch it. It's really cool. It really kind of gives you a better visual as to why we call the cell membrane or the plasma membrane. Those two words are used interchangeably. And why we call it a fluid mosaic model. Inside the membrane, we have our cholesterol molecules. I always like to call it a fluidity buffer because it prevents changes or some significant changes with the membrane being too runny and then not being able to keep its contents inside and stuff outside that we don't want in, or too thick because we do want things to go in and out. So the cell will literally alter the amount of cholesterol in there to maintain the proper fluidity. We have membranes inside that, uh, or proteins inside that membrane called integral proteins and peripheral. So the integral proteins, they enter the hydrophobic area. So you can see here, they're in here. Okay. Now, number one right here is called a transmembrane because it goes all the way across. All right. Then we have 2 and 4, which is here and here. They are called integral proteins just because they penetrate, they go down into those hydrophobic tails. And then we also have peripheral. Peripheral would be this one up here. It just kind of hangs out with those hydrophilic heads, whether it's on the outside or the inside of the cell. Okay. So you'd have the same situation just with it down, maybe right in through here. Okay. So those are called peripheral proteins. So we can see all the stuff going on in that cell membrane. Lots and lots of things. These are all like showing you different types of proteins with the alpha helixes and beta pleated sheets. When we talked about structures of proteins, we can see some proteins that are going across, some other types of proteins, more like cytoskeleton. So a lot of things are going on in that cell membrane. There are also different types of proteins inside that cell membrane that has a lot of different functions, whether it's transport, enzymatic, signal transduction, cell-to-cell recognition, intracellular joining, or attachment. And wow, I can count, or it's that auto stuff. There's 6. Okay. So I like this picture because it shows a lot of these proteins here. It shows transport, specific ones letting things in and maybe out. This one in particular requires energy for that type of carrier protein right there to work. They don't all need energy, and we'll get into that later in this chapter. We also have receptor for transduction, meaning that this molecule, which is often called a ligand, we talked about enzymes and how enzymes bind to substrates. When we talk about other types of receptors and stuff like that, we generally call it a ligand. And so that ligand attaches, it usually kind of causes some kind of shape in that protein, and then it relays the message in. So that ligand does not go into the cell. It's just um, passing off a message, you could say. We also have some that anchor. We're going to anchor the cytoskeleton here and that ECM on the outside. We've got enzymes. We talked a little bit about enzymes and did our enzyme lab. And then we have intercellular joining. So we also have some that are just going to help keep those cells connected to one another. And then we have cell to cell recognition. And a lot of this is used so that cells can recognize each other so they know it's not like a bacterial cell, in which case it would elicit an immune response. So membrane permeability, molecules that are nonpolar can dissolve in the lipid bilayer and therefore can cross the cell membrane. Small molecules are also able to just kind of zip through there. Large or polar, they have a very difficult time getting through the cell membrane. Therefore, they're going to need help. And that help is generally with the in with the protein facilitating that transport into and out of the cell. Here is water. Even though water is small and it can zip through. We don't often get enough of it either in or out, depending on the situation of the cell. And so we do have what are called transport channels. And this one is called an aquaporin because it specifically moves water. Water will move according to its concentration gradient. So where it's high, it's going to go low. Where it's low, it's so whether it's high outside the cell, it may lose water. If it's low inside the cell, then it's going to bring it in. I like this diagram because it shows you like right here that cohesion, that hydrogen bonding we call cohesion. And then right here is adhesion. Oh, excuse me. We talked about that with hydrogen bonding. So we can kind of review that concept there. Here is a carrier protein. This is the sodium potassium pump. Oops, sorry. My bad. It's the it's a symport for glucose. Okay. So when we talk about symports, that means we have two molecules moving in the same direction. Remember, sim means with in Latin. Okay. So we have the potassium moving in here. Oh, that one's sodium potassium. There, see, I'm not losing it. I thought so. So this one here is a sodium potassium pump, and I'm going to talk about that one in a lot more detail. It's kind of very important for you guys to know. But this one's that symport I was talking about. So you have the sodium that comes in and it's going to bring glucose, and then it's going to change shape and it's going to let go of the sodium and the glucose. So we always talk about how sodium chloride salt is bad for you, but we actually need, there's a lot of ions that we really do need. We need sodium, we need potassium, we need calcium. So these are often called electrolytes. So when you guys are learning about and when you do exercise and you start cramping up and whatnot, it's generally because you're low on your electrolytes and sugar, which is why Powerade and Gatorade and all that are full of electrolytes and sugar to help replenish your body from high exerting activities. All right, so that's just kind of like the beginning of this chapter. There's not a whole lot to the beginning, but then we're gonna get into passive and active transport, which is it's gonna be it's definitely gonna be harder than this first part of the chapter. So I'm gonna go ahead and end this video, and I will see you in the next video, which again, it's gonna be a little more complicated. All right. Thanks for watching, and I will see you in a second.